Hi everyone. In this video, we're going to look at genetic recombination and linked genes. Thomas Hunt Morgan once again selected Drosophila melanogaster as his model organism for studying how gene linkage, that is, genes that are on the same chromosome, would affect inheritance patterns. He started with Drosophila with either gray bodies and long wings, the wild-type phenotypes, and crossed them with a double mutant with black bodies and vestigial or short wings. The F1 offspring then would all be heterozygous for both body color and wing size. He then performed a test cross by breeding the F1 dihybrids with the double mutant to determine the outcome in the F2s. Now, Morgan hypothesized that if the genes were on different chromosomes, then independent assortment would apply. And there would be an equal number of offspring with each of the four possible combinations. The two parental varieties with either gray bodies and long wings, or black bodies and vestigial wings, as well as the recombinant phenotypes, that is, the combinations that weren't found in either parent. Now, he also hypothesized that if the genes were on the same chromosome, then only the parental types would be seen and there would be no mixing and matching. What he found was actually neither of those hypotheses were supported. In the more than 2,200 flies that he studied, the parental varieties were found in the majority of the offspring, but there were a much smaller number of recombinant offspring. So let's look at a quick recap of how genetic recombination can mix and match genes that are on the same chromosome. We start with our homologous pair of chromosomes, which, remember, come together during prophase 1 of meiosis. Now, since we're in meiosis 1, each chromosome is comprised of two sister chromatids, which at this point are identical to each other since they were produced by DNA replication during S phase. And when the homologous pair comes together like this, the non-sister chromatids, that is, one chromatid from each chromosome, can cross at regions called chiasmata. Where that occurs, chromatids will break and rejoin, exchanging ends with each other, resulting in chromatids carrying new combinations of alleles that weren't found in either parent. So let's look at how recombination happened in our flies. And we're going to start here at the F1 generation, since the parental generation were homozygous. In most of the cases, the body color and wing size genes remain together, resulting in F2 offspring with the same genotype as one of their parents or the other. However, in some of the cases, there was a crossing over event that separated the genes, resulting in offspring with a combination of parental phenotypes. These are what we call the recombinant types. We can use the numbers of offspring with each phenotype to determine the frequency of this particular recombination event by dividing the number of recombinants by the total number of offspring and multiplying the answer by 100, which in this case gives us a recombination frequency of 17%. Now, the further apart the genes are on a given chromosome, the more likely they will be separated by recombination, and so the higher the frequency will be, until we reach 50%. Genes that are very far apart on a chromosome will essentially behave as if they are on separate chromosomes, and so will be separated approximately 50% of the time, just as they would if they underwent independent assortment. The recombination frequency cannot go over 50%, even if the crossing over is guaranteed to happen somewhere in the space between the genes, because there are always two chromatids that didn't undergo recombination for every two that did. So the genes at opposite ends of the chromosome, while physically linked, will behave as though they are genetically unlinked and will show the same frequency that we would see from independent assortment. Thanks for watching, and if you haven't had a chance to check it out yet, please go ahead and watch the video on X-linked inheritance.